Good day, everyone. I am Rosita C. Mabolo, and today I'm going to present to you my topic, the Clostridium perfringens. So this would be the outline of my presentation. This would cover the physiology and structure, pathogenesis and immunity, epidemiology, clinical diseases, laboratory diagnosis, and treatment control and prevention. Let's start with its physiology and structure. So, Clostridia are gram-positive, anaerobic, endospore-forming bacteria and comprising approximately 180 species. The genus Clostridium is one of the largest bacterial genera. And for Clostridium perfringens, it is a gram-positive, spore-forming, strictly anaerobic bacterium that is ubiquitous in nature, being found in variety of locations such as soil and gastrointestinal tract of mammals, and it is a large, rectangular, and with spores rarely observed either in vivo or after in vitro cultivation, which is an important characteristics that differentiate this species from most other Clostridia. The colonies of Clostridium perfringens are also distinctive, with a rapid spreading growth in the laboratory media and B hemolysis on blood-containing media. The production of one or more major lethal toxin by Clostridium perfringens such as alpha, beta, epsilon, and iota toxins is used to subdivide isolates into 5 types A through E. Let's take a look into the alpha toxin. So alpha toxin is responsible for a gas gangrene and myonecrosis in infected tissues. The toxin also possesses a hemolytic activity and has been shown to be the key virulence factor in infection with Clostridium perfringens. The bacterium is unable to cause a disease without this toxin. And the alpha toxin is a zinc methanol phospholipase requiring zinc for activation. So first, the toxin binds to a binding site on the cell surface. The C-terminal domain binds calcium and allows the toxin to bind the phospholipid head group onto the cell surface. The C-terminal domain enters the phospholipid bilayer, and then the N-terminal domain has a phospholipase activity. This property allows hydrolysis of phospholipids such as phosphatidylcholine, mimicking the endogenous phospholipase C. The hydrolysis of phosphatidylcholine choline produces a diacylglycerol, which activates a variety of second messenger pathways. The end result includes the activation of arachidonic acid pathway and production of thromboxane and production of the IL-8 platelet activating factor and several intercellular adhesion molecules. These actions combine to cause edema due to increased vascular permeability. So the second one, we have the beta toxin. So the beta toxin is one of the 4 major lethal protein toxins produced by the Clostridium perfringens type B and type C strains. It is a necrotizing agent and it induces a hypertension by release of catecholamine. It has been shown to cause a necrotic enteritis in mammals and induces necrotizing intestinal lesions in the rabbit ideal loop model. So the Clostridium perfringens beta toxin is susceptible to breakdown of proteolytic enzymes, particularly the trypsin. The beta toxin is therefore highly lethal to infant mammals because of trypsin inhibitors present in the colostrum. So for the pathway involved, using a standard settings, initial binding of the CPB or the beta toxin to its potential receptor, the ATP-gated P2X7 receptor, induces a rapid peak of ATP release from target cells. And then this ATP loss is not associated with cell lysis and it would occur through the ATP release channel pannexin 1. And then the released ATP would stimulate further beta toxin binding and oligomer formation, facilitating the pore forming activity of the toxin. Pore formation results in the calcium 2 plus influx and loss of an enterocytoplasmic K plus IK plus, and then the increase of the calcium 2 plus is associated with the calpain activation and necroptosis, which is inhibited by the NEC1. Only low levels of caspase 3 activation occurs, suggesting that apoptosis is not a significant mechanism of cell death. So the decrease of IK plus is associated with the activation of the MAPK and JNK, which activate host cell survival and defense pathway. So the dash or a dash block arrow shows direction of movement, as you can see in the picture reflected in your screen. Third one, we have the epsilon toxin or ETX, which is produced by Clostridium perfringens type B and type D strains and causes enterotoxemia, a highly lethal disease with major impacts on the farming domestic ruminants, particularly the sheep. So the epsilon or ETX belong to the aerolysin-like pore-forming toxin family. And so for its pathway, the epsilon toxin binds to the plasma membrane of sensitive cells and forms an oligomer required for pore formation in plasma membrane lipid rafts. And then the lipid composition of the membrane and the membrane fluidity have been shown to be a critical factor for oligomerization and pore formation by epsilon toxin. Ceramide production in the plasma membrane results in the ceramide clustering, thereby initiating the formation of microdomains. One, we have the iota toxin, which is produced by the type E strains of Clostridium perfringens. It has a natriuretic activity and increases vascular permeability. So the Clostridium perfringens iota toxin is composed of the enzyme component IA and the binding component IB. So what happened in here is that IB binds to a receptor or targeted cells and translocates IA into the cytosol of the cells. IAAGP ribosylates actin, resulting in cell rounding and death. Comparisons of the deduced amino acid sequence from the gene and three-dimensional structure of IA with those AGP ribosylating toxins or ARTS suggests that there is a striking structural similarity among these toxins. We also have the Clostridium enterotoxin or the CPE, which is responsible for causing the gastrointestinal symptoms of several Clostridium perfringens food and non-food-borne human gastrointestinal diseases. The enterotoxin gene CPE is located on either the chromosome or a conjugative plasmids. In all CPE-positive strains, the CPE gene is strongly associated with insertion sequences that may help to assess its mobilization and spread. So during the disease, the CPE is produced when the Clostridium perfringens sporulates in the intestines, a process involving several sporulation-specific alternative sigma factors. And then the action of the CPE starts with its binding to claudin receptor to form a small complex. Those small complexes then oligomerize to create a hexameric prepore on the membrane surface. The beta hairpin loops from the CPA molecules in the prepore assemble into the beta barrier that inserts into the membrane to form an active pore that enhances calcium influx, causing a cell death. This cell death results in intestinal damage that causes the fluid and electrolyte loss. And then the CPA is now being explored for translational applications, including the cancer therapy or diagnosis, drug delivery, and vaccination. So for its pathology, Clostridium perfringens is not highly invasive and requires damage in the tissue. Conditions stimulate spore germination, vegetative growth, and other violence factors, and fermentation of muscle carbohydrates result in the formation of gas and further destruction tissue. So we are now on the epidemiology. The Clostridium perfringens bacteria are one of the most common causes of a foodborne illness or food poisoning. The CDC, or the Center for Disease Control and Prevention, estimates that these bacteria cause nearly 1 million illnesses in the United States every year. Local, state, and territorial health department voluntarily report Clostridium perfringens outbreaks to the United States. The type A Clostridium perfringens commonly inhabits the intestinal tract of humans and animals and is widely distributed in nature, particularly in soil and water contaminated with feces. Spores the spores form under adverse environment conditions and survive for prolonged periods. Strains of type B through E do not survive in soil but colonize the intestinal tracts of animals and occasionally humans. Clostridium perfringens particular type A is responsible for a variety of diseases including soft tissue infections, food poisoning, a necrotizing enteritis, and asepticemia. So for clinical diseases, first one we have the cellulitis. Is a common bacterial skin infection that causes redness, swelling, and pain in the infected area of the skin. If untreated, it can spread and cause serious health problems. Different types of bacteria can cause a cellulitis, which is an infection of the deeper layers of the skin. Experts do not know how the bacteria get into the body for many people who get cellulitis. Sometimes the bacteria get into the body through openings in the skin, like an injury or surgical wound. So in general, people cannot catch cellulitis from someone else. Anyone can get cellulitis, but some factors can increase the risk of getting this infection. Um, we have here the risk factors because they allow bacteria to get through the skin. So first, we have the injuries that cause a break in the skin, like cuts, ulcers, bites, puncture wounds, tattoos, and piercings. And then the chronic skin conditions like athlete's foot and eczema, and then chickenpox, and then injection drug use. Then we have also the fasciitis. It is a rapidly progressive bacterial infection that tracks along the fascia and associated with severe sepsis and deadly prognosis. The diagnosis is rare compared to cellulitis and abscesses, though it may initially present very similarly, making recognition a challenge. Several types of necrotizing fasciitis exist and differ by microbiology and typical affected population. So fasciitis is a rare bacterial infection that spreads quickly in the body and can cause death. The bacteria most commonly enter the body through a break in the skin, including the cuts and scrapes, burns, insect bites, puncture wounds, including those due to the intravenous or for drug use, and then surgical wounds as well. And then we have the gas gangrene. It is a life-threatening disease that illustrates the full virulence potential of histotoxic Clostridia. The onset of this disease is characterized by intense pain and generally develops within a week after Clostridia are introduced into the tissue or by trauma or surgery. The onset is followed rapidly by extensive muscle necrosis, shock, a renal failure, and death, and often within 2 days of initial onset. The microscopic examination of muscle reveals a devitalized necrotic tissue. Gas found in the tissue is caused by the metabolic activity of the rapidly dividing bacteria. So that is why nga gitawag ni siya og kanang gas gangrene because of the gas nga makita sa tissue. So a strain of tissue is exudate collected from the wound of a patient with Clostridium perfringens necrosis will reveal abundant rectangular gram-positive rods with absence of inflammatory cells resulting from lysis by Clostridial toxin. So the clostridial toxins characteristically cause extensive hemolysis and bleeding. So for clostridial food poisoning, it is a relatively common but underappreciated intoxication. It's characterized by short incubation period, 8 to 12 hours, and clinical presentation that includes abdominal cramps and watery diarrhea but no fever, nausea or vomiting, and a clinical course lasting less than 24 hours. So the disease result from ingestion of meat products, example are beef, chicken, and others, contaminated with large numbers, like for example 108 to 109 organisms of enterotoxin producing type A Clostridium perfringens. So the holding contaminated foods at temperature less than 60 degrees Celsius allows spores that survive the cooking process to germinate and multiply to high numbers. And then rapid refrigeration of food after preparation prevents this bacterial growth. Alternatively, reheating the food to 74 degrees Celsius can destroy the heat level enterotoxin. So for necrotizing enteritis, it is a rare necrotizing process in the jejunum characterized by acute abdominal pain, vomiting, bloody diarrhea, ulceration of the small intestine, and perforation of the intestinal wall, leading to a peritonitis and shock. Mortality in patients with this infection approaches 50%. The beta toxin produced by Clostridium perfringens type C is responsible for this disease. So for laboratory diagnosis, most people with Clostridium perfringens do not receive a diagnosis immediately. Infection is confirmed when a laboratory test detects a bacteria or other toxin in a sample or in a stool sample, or the bacteria found in the food linked to illness. The laboratory performs a confirmatory role in the diagnosis of clostridial soft tissue diseases because therapy must be initiated immediately. The microscopic detection of gram-positive rods in clinical specimen, usually in the absence of leukocytes, can be a very useful finding because these organisms have a characteristic morphology. It is also relatively simple to culture these anaerobes. Under appropriate conditions, Clostridium perfringens divide every 8 to 10 minutes, so growth on agar media or blood culture growth can be detected after incubation for only a few hours. And also, immunoassays have been developed for detection of the enterotoxin in fecal specimens. However, clostridial food poisoning is primarily a clinical diagnosis, and culture for immunoassays are not commonly used. So for the treatment, control, and prevention, immediate cleansing of dirty wounds, deep wounds, compound fractures, and infected incision is necessary. Debridement of diseased tissue is also needed. So if we say debridement, it is a procedure for treating a wound in the skin. It involves thoroughly cleaning the wound and removing all hyperkeratotic or thickened skin or callus, infected and non-viable tissue, foreign debris, and residual material from a dressing. So Clostridium perfringens soft tissue infections such as suppurative myositis and myonecrosis must be treated aggressively with surgical debridement and high-dose penicillin therapy. Hyperbaric oxygen treatment has been used to manage these infections as well. However, the results are inconclusive. Despite all therapeutic efforts, the prognosis in patients with this disease is poor, with mortality reported from 40% to 100%. Less serious localized soft tissue infections can be successfully treated with debridement and penicillin. Clostridial food poisoning is managed by oral rehydration, and in severe cases is a uh, intravenous fluids and electrolytes. Antibiotics therapy is not recommended because this is a self-limiting diseases. For example, the diarrhea washes the bacteria out of the intestine and the normal intestine flora is reestablish itself. And then exposure to the Clostridium perfringens is difficult to avoid because the organism are ubiquitous, and then the disease requires introduction of the organism into the vitalized tissues and maintenance of an anaerobic environment favorable for bacterial growth. Those proper wound care and judicious use of a prophylactic antibiotics can do much to prevent most infections. And that brings us to the end of my presentation. Thank you so much for listening.